Hello, good morning, everyone. Hope all of you are doing well. So today, our agenda is to do the Lecture 5 study guide. It's actually quite a difficult concept, especially if you are learning it for the first time. So I will go through it in more details, repeat a little bit of what I said in the lecture recording as well. And if we have time, we will take up the lab and also some of the questions from quiz number 2. Okay? But if we don't have time for those, then we'll do it during the review class for the test. Next week, there is no class because it's engagement week. You have no class for all of your subjects. It's essentially kind of like a week off, if you would. And then when we come back, we'll have a test number 1. Okay, so the tutorial is going to be on the Monday instead of the Wednesday when we come back. I'll post all these information online afterwards, and now I'll talk about it again, before we end, so there's no confusion. All right. So here we go. Metabolism. First question here. Writes the word equation and a balanced chemical equation for cellular respiration. Indicate which reactant is oxidized and which is reduced. So let's start with our word equation for cellular respiration. We begin with glucose. Okay, glucose. And we are going to react it with oxygen. And in the process, we will be, producing carbon dioxide and water. Okay. And most importantly, we are going to be creating ATP, the adenosine triphosphate, which is the usable form of energy for our cells. Okay. So in this reaction, the glucose is going to be the one that's being oxidized. Okay, it is the one that is going to be releasing oxygen, releasing electrons. Okay, so glucose will be oxidized into carbon dioxide. Okay, so loss of electron equals to oxidation. Okay, so you can remember it as like Leo, right? Loss of electron is oxidation. Okay, so all those electrons are going to be very high in energy, and we will be capturing those electrons along the way. Okay? Ultimately, the electrons will be transferred to oxygen. Okay? So oxygen is going to be reduced into water. Okay? So this is reduced. Okay? So when something gains electron, we call it reduction. So you can remember it as GER. Gaining electron is reduction. Okay. So of course, this is not going to be happening in a single step. If you have watched the recordings, you will know this happened in 4 major stages, and each stage is going to have multiple steps in it. Okay, but this is the ultimate equation that summarizes, the reactants and the products. So that's a word equation. We can write a chemical equation. The formula for glucose is C6H12O6. We are going to be reacting it with 6 oxygen. Okay, as you learn in chemistry class, oxygen is always going to be diatomic. It's always going to be with a 2. Producing 6 carbon dioxide, 6 water, and up to 36 ATP molecules. Okay, so that is the chemical equation. All right. So let's do a quick summary of what's going on in this unit. You should maybe write this down. You should definitely write this down, and you know, ask questions if you have any. Okay, so this is just metabolism. Okay, this is like the condensed version of what the PowerPoint has. Okay, so we're talking about cellular respiration. Okay, which is a type of metabolism, right? Last time we learned that metabolism can be anabolic or catabolic, depending on whether you are building things or breaking things down. So what do you guys think? Cellular respiration, is that anabolic or catabolic? Which type would it be? Anybody? Yeah, this is gonna be catabolic, okay? Catabolic reaction, because we're breaking things down, we're breaking down glucose, and we're gonna be releasing energy along the way. Okay, so this can be thought of as catabolism. Okay, breaking down glucose. Okay, so there are 4 major steps that are involved. Right, we already saw the overall equation, right? So what are the 4 major steps? Anybody remember it from the lecture recordings? What's the first step in cellular respiration? Yeah, so many people say glycolysis. Okay, so it's the splitting of glucose. Lysis is the break. Glyco is talking about the glucose. And then we're going to have pyruvate oxidation. Right? We're going to strip more electrons from the pyruvate. And then after that, we're going to have the Krebs cycle. And then finally, the electron transport chain, okay, or the ETC. So essentially what's going to be happening is these 3 steps, the first 3 steps, the glycolysis, pyruvate oxidation, and the Krebs cycle, you will be releasing high energy electron as glucose is being oxidized. And remember, to be oxidized means you're stripping electron off something, right? So as we remove the electron from the glucose, right, in these 3 steps, the high energy electrons will be released. Okay, so we want to capture these high energy electrons, okay? And to capture it, we have 2 energy carriers, right? Anybody remember the names of the energy carrier that we have in the body? The short form is fine. You don't have to tell me the full name. Energy carrier. Okay, thank you, Tina. There is the NAD plus, and then there is the FAD. Okay, so these are, for those of you who need a refresher, these are energy, or you can think of them as electron carriers. Okay, electron carriers. Okay, as the name implies, they carry electrons. Okay, so all these electrons will be released from the first 3 steps, and then they will be picked up by these energy carriers. So NAD plus will pick up the electron, and then it will become NADH. Okay, so you know, you add some electrons to it, and then same thing here. FAD, if you add some electrons to it, then it will become FADH2. Okay, I'm simplifying here. It's more complicated than that, but that's all you need to know. All right. NAD plus, FAD, they are the empty version Once you fill them up with electron, they become the charged version, the full version, the NADH and the FADH2. Does anybody have any question about what these are? It's essential you understand what they are because we'll be talking about them the whole class. Okay, we'll be doing calculations with them. So any questions about what I'm saying before we proceed? We all good here? No questions. Okay. So in the lecture, I said think of these as like taxis, right? Okay, these are like unoccupied taxis, and then the electrons are like the passengers. Once the passenger gets on, it becomes occupied. Okay, so where are we taking these electrons? Okay, we will be taking the electrons all the way to the ETC. Okay, where the energy from electrons will be used to produce lots of ATP. Okay, so once the NADH deposit the electron at the electron transport chain, it will once again become NAD plus, and then it could go back and pick up more. Okay, so NAD plus is basically like a ferrying system. It goes back and forth, grabbing more electrons, bringing to ETC, grabbing more electrons, bringing to ETC. Same thing for FAD. They are both energy carriers. So the difference, can someone tell me what the difference is? What's the difference between NAD plus and FAD? Anybody want to unmute and tell me about that? What's the difference?
The NAD plus carry 3 ATP, but the FAD just carry 2 ATP.
Okay, yeah. So that's pretty good. But I'm gonna make a small adjustment to what you said. What she said is correct. Okay, but they're not carrying ATP. Okay, they are carrying high energy electrons. Okay, so NAD plus will carry enough energy to produce up to 3 ATP. Okay, whereas FAD, they will carry enough energy to produce up to 2 ATPs. All right, so just to clarify, these are carrying high energy electrons. They're not carrying ATP. Okay, but the electrons that they carry will be used to produce ATPs. Okay, so the difference here is NADH carries enough energy to make up to 3 ATP. If you did not know that, you definitely should write that down. Whereas FADH2 carries enough energy to make up to, what did I say, 2 ATPs. Okay, so that's one key difference. All right, think of them as like batteries, I guess. Okay, so this is like your 9-volt battery, the rectangle ones. It holds more charge than, let's say, your AA batteries. Okay. All right. So where is this happening, right? Where is this happening? Here is your cell. Here's your cell. This is the cytoplasm. Okay, and then we have our mitochondria, right? Okay, so step one is glycolysis. Glycolysis will take the glucose and then it will split it into 2 pyruvate. Okay. And then the 2 pyruvate will go into the matrix. Okay, so in the matrix, number 2 and 3 will happen. The pyruvate oxidation will happen in the matrix. The Krebs cycle will also happen in the matrix. And finally, the ETC, the electron transport chain, will happen in the cristae. Remember the foldings? That's called the cristae, right? Okay. So let's go back here and try to fill in this chart. stages in cellular respiration and you know what they produce. So again, the first step is glycolysis. Where does it take place? I just told you, it's in the cytoplasm. Okay, so the key things that will produce would be 2-pyruvate. If you read it in a textbook or like in an online textbook or you know anywhere else, sometimes they may say pyruvic acid, and that's okay. Okay, both are the same thing for our purpose. They're not really the same thing, but doesn't really matter for us. Okay. Now you are also going to be producing 2 net ATP. I'll explain what that means in a second. So 2 ATP net, as well as 2 NADH. Okay. So let's go into a little bit more detail explaining the glycolysis. So like I said, glycolysis begins with glycolysis. That begins with 1 glucose, right? So glucose. And glucose has 6 carbons in it, right? C6H12O6. Okay. And we are actually going to undergo a series of reaction which will ultimately split the glucose into 2 pyruvate. So pyruvate and pyruvate. So let's do some simple math here. If we started with 6 glucose, after you split it into 2 pyruvate, how many carbons do we have for each pyruvate? Yeah, thank you, Tyler. So that's 3 and 3. Okay. Now for glycolysis, it is separated into 2 separate phase, we would say. Okay, so we have number 1, we have the energy investing phase. Okay, and then with number 2, we have the energy yielding phase. Okay, so you might have heard of people say, you know, it takes money to make money. Okay, you invest $100 into I don't know, like into the bank, okay, into some investment, okay, and then you might get $104 back after a year, okay, if they pay you 4% interest. Okay, so you have to invest a little bit before you get some back, right? So this is essentially what happens in the energy investing phase. You are going to basically use 2 ATPs, okay, to energize glucose, to make it unstable, right? Okay, to make it want to react. All right. I don't know if you remember this, but I definitely talked about it in lecture 2. Remember, in a reaction, right? In order for a reaction to occur, you must supply what we call the activation energy, right? That's the energy you need to invest initially to kickstart the reaction, right? Before it's gonna happen. Right? So that's what we're talking about here. The 2 ATP will energize the glucose, make it unstable, make it willing to react. Okay, so that's going to be like the first 5 steps in glycolysis. Glycolysis has a total of 10 steps, right? So in the energy yielding phase, that's where the energy you invested will pay off. Okay, you will get 4 ATP back. 4 ATPs will be produced. Okay, so that's why it's a net production of 2 ATP. Okay, so 4 ATP minus 2 ATP gives you 2. In addition, we will be releasing some electrons along the way, and those will be captured by NAD+. So 2 NAD+ will come, and after capturing the electrons, you will produce 2 NADH as well. Okay, or generated, produced, whatever. We'll just use the same word. Okay, so that's what you invest and that's what you produce. Okay, Now I'm going to put a star here. I'm going to put a star here. Okay. These ATPs are produced by what we call substrate level phosphorylation. Okay. You know what, guys? I'm gonna put this stuff on the other page. Otherwise, we will keep on running out of space. Sorry about that. Cut. And we'll paste it right here. Let's see if it's gonna work. Oh, it does work. Very good. Okay. So the four ATPs that are produced in the energy yielding phase of glycolysis is going to be produced by a process called substrate level phosphorylation. Now that's a big word. Let's break it down and see what that means. Okay, phosphorylation. To phosphorylate something is to put a phosphate on them. Okay, so to phosphorylate something is to add a phosphate to a molecule. A phosphate is PO4, right? -3. You probably know that from chemistry. Okay, so in this case, substrate level phosphorylation is going to involve three things. Okay, we are going to have, you know what? I'm just going to grab the picture from the lecture notes. That way, I don't have to redraw it. There we go. Okay, so this is what substrate level phosphorylation looks like. Right? You're going to have an enzyme to catalyze the reaction. We're going to have this donor molecule or fuel or donor molecule. That's the molecule that you will be ripping the phosphate off. Okay, so you see this is a very good molecule. It has 2 phosphate. So the enzyme will rip off one of the phosphate and then it will transfer the phosphate to ADP. So when you have ADP plus a phosphate, we talked about this last time when we had a tutorial, that will become an ATP. Okay, so in this case, we are phosphorylating, adding a P onto ADP and therefore creating ATP. And you can see our donor molecule, after you rip the P off, there is only 1 P left. Okay, so this process is called substrate level phosphorylation. Okay, very small percentage of ATP is produced this way. Okay, we'll say approximately 11% of all ATPs are produced using substrate level phosphorylation. I'm talking about the entire cellular respiration. Okay. Are we together? You guys are awfully quiet. Any question about what I've said? Good. Thumbs up. Happy? All right. So what's going to happen next? The suspense. What happens next is the pyruvate, the 2 pyruvate that we just produced. Remember, each of them contains 3 carbons, right? That is going to go into the matrix. Okay, they're going to head into the matrix, which is the innermost portion of the mitochondria, right? Okay, so step number 2. Step number 2 in the matrix. Now we're going to do pyruvate oxidation. Okay, so in here, 3 things is going to happen. Okay, the pyruvate, which is 3 carbons. Okay, we are going to remove a carbon from pyruvate, and it's going to come out in the form of carbon dioxide. Okay. We are also going to be releasing electrons. So an NAD plus will come along, it will grab the electron, and then it will become NADH. Where is the NADH going to go afterwards? Let's take a moment and think about what we said earlier. Okay. The NADH will grab, after it picks up the electron, it's going to go to ETC, okay, the electron transport chain, the last step where it will deposit the electron so that it can make 3 ATPs, right? Okay, and also we're gonna add an so let's just do it like this. We're gonna add something called coenzyme A, which like charge up the molecule. We create a special unstable bond that will charge up the molecule so that it is ready for the next phase of the reaction. Okay, so in all this, we will create something called acetyl-CoA. Okay, so how many carbons would be in the acetyl-CoA? How many carbons from the original glucose, I would say, at this point? That's something I could ask you on the test, right, which is what I'm telling you. Okay, how many carbons? My question is, we started with 6 carbons, right? For the glucose, you split it into 2 pyruvate, so 3 carbon, 3 carbon, and then now we remove a carbon dioxide, right? So yeah, we're gonna have 2 carbons left in the acetyl-CoA. Okay, 2 of the carbons from the original glucose. Okay, because you take away the carbon dioxide, right? That's why like 3 minus 1 gives you 2. Yes. All right. Now listen to me carefully and think about it. How many times does this reaction takes place for every glucose that we break down? How many times does this reaction happen for every glucose that we metabolize? That's right. Okay, people are saying 2 times, twice. That's correct. Remember, each glucose is broken down into 2 pyruvate, right? So each of the pyruvate will do this once. Okay, so per glucose, this happens twice. So if we go back to here, we can fill that in. So step number 2 is going to be pyruvate oxidation. You're going to be oxidizing the pyruvate. We're going to be stripping electrons from it. And this happens in the matrix, the innermost portion of the mitochondria. And we are going to get 2 carbon dioxide from this per glucose. We're going to get 2 NADH, and we're going to get 2 acetyl-CoA. Okay, remember, we're adding the coenzyme A to supercharge the molecule so that it is ready for the next step. All right, so I'm just going to put a note here. All this is per glucose. All right. I know I keep on asking if you guys have any questions, but you guys are good, right? Okay, please let me know if you do. Okay, it's very important that you do. Okay, let's see if we can do some polls here. Yeah, let's do some polls just to, make sure you're not dozing off or anything. First question, let's go. All right. I'm going to end the polls, even though only half of you responded. The correct answer is pyruvate. Okay, pyruvate. All right. So NADH, right? Let's think about this for a second. NADH can only produce 3 ATP, right? Okay, up to 3 ATP. Carbon dioxide is a waste. It has no energy that we can use. ATP is just 1 ATP. But what about the pyruvate? Well, based on everything we learned so far, let's see what happens. The pyruvate, when it goes on to do the second step, it can produce an additional 2 NADH. Okay, so just up to this point, and we haven't even talked about what happens in step 3, right? Up to this point, we already squeeze out more energy from pyruvate than we would have got from 1 NADH, right? Okay, so you can see at the end of glycolysis, the pyruvate still has the most chemical energy. Okay, so this will continue to be broken down to produce many more ATP, NADH, and FADH2. Okay, so that's why that's the one that has the most chemical energy after glycolysis. All right. This is an important one to know. Okay. We got to talk more before we go on. So that is the pyruvate oxidation. Let's go to step number 3. Okay, step number 3. What is going on in step number 3? Step number 3 is the Krebs cycle. So number 3, Krebs cycle, KREB cycle. Okay. So what happens is the acetyl-CoA will come in. Okay. And in the matrix, we're in the matrix right now. In the matrix, there is going to be a molecule called oxaloacetate. Now, this oxaloacetate is going to have 4 carbons in it. Okay, you just have to believe me. So when you take the acetyl-CoA and the oxaloacetate and you combine it, you're going to end up with 6 carbon, right? So this 6 carbon molecule is citrate. Okay, citrate. Okay. So which is why the Krebs cycle is also called the citric acid cycle, right? Okay. So what's going to happen here? Well, the citrate, it is going to be broken down. It is going to be metabolized in several steps. We're not going to worry about what those steps are, but suffice to say, you are going to eventually regenerate the oxaloacetate, and along the way, you will be releasing 2 carbon dioxide. Okay, so let's see, if you take 6 carbon and then you minus 2 carbon, you get back the 4 carbon here, right? Okay, and also along the way, you will be releasing high energy electron, and some of it will be captured by the NAD+, and you will generate NADH. So that's going to be a total of 3 NADH. And then we will have an FADH as well. Okay. And also we will be producing 1 ATP. Okay. So that is what happens in 1 spin of the Krebs cycle. Okay. These are all the things that you'll be producing. Okay. So of course, these guys, they are going to go to ETC, right? And this is going to be released out of the body. You breathe it out. Okay, and this is just ready to be used. If you need ATP for whatever reason, you can use it right away. Okay, so that is the Krebs cycle, the key events anyways. So when we go back to here, we can fill it in. Okay. Krebs cycle. Also taking place in the matrix. Okay. Now, just going to go back to show you here one more time. Okay, we're going to have, oh, sorry, these things are like so big. It's hard to, let me just make it smaller. There you go. Okay, so we're going to have 2 carbon dioxide, 3 NADH, 1 FADH, and 1 ATP, right? And that's for one spin of the cycle. Remember, we have 2 acetyl-CoA coming in, right? So on a per glucose basis, right, the Krebs cycle will produce 4 carbon dioxide, 6 NADH, 2 FADH2, and 2 ATPs. Okay. So these ATPs, I'm gonna put an asterisk here. These ATPs, right here, produced through substrate level phosphorylation. That's the one where you rip the P from a donor and then you pass it on to ADP. All right. So you can see the carbon dioxide, right? 2 carbon dioxide here, 4 carbon dioxide there. It makes up a total of the 6 carbon dioxide that we see over here, right? 6 carbon dioxide right here, right? Okay, so these carbons that are in the carbon dioxide, those are the same carbon that was found originally in the glucose. Okay, and then in step 4, which I'll explain in more details in a second, but step 4 is going to be the electron transport chain that happens in the cristae, and the little foldings of the inner membrane. And that will produce up to 32 ATP. We're going to be regenerating some of the NAD+. We're going to be regenerating FAD. So these could go back to the first 3 steps to pick up even more electrons for the next round. And we will be producing 6 H2O. Okay, so of course, these H2O corresponds to these H2O. Okay, so remember, the oxygen will get reduced into the water, whereas the glucose will get oxidized into carbon dioxide. Okay, so oxidize, just like what we said above, and then this is going to be reduced. Is that okay? All right. So I will put in other stars. There's so many stars going on here. Put in other stars right here. Okay, so these are produced. These ATPs in the ETC, they are not produced through substrate level phosphorylation. They are produced through oxidative phosphorylation. Okay, which I'll explain right now. And again, right, just in case you confuse about these big words. To phosphorylate is to put a P onto something. We're trying to make ATP, so we are trying to phosphorylate ADP, right? So ADP plus a P, that creates ATP. Okay, so when you add the P onto ADP, is called phosphorylation, right, of ADP. How are we phosphorylating it? Are we phosphorylating it using an enzyme and a substrate and a donor? If we are, then it's called SLP. That happens in the first step and the third step. If we are phosphorylating ADP using oxidation reaction, right, then it is called oxidative phosphorylation, which is what's happening in the last step. Okay, so here we go. Let's draw this thing out. It's already explained in the lecture, right? Like I said, but I know you guys have been having a busy week with all the tests and stuff, so maybe you didn't have a time to have time to watch it yet. So I'll give you a condensed version of it. Okay, a summary. So this is the outer membrane, outer mitochondrial membrane. And then we have the cristae here, right? The inner membrane, the cristae. This is the matrix. This is the intermembrane space. And then out here, we have the cytoplasm. Okay, so we're basically zooming onto the mitochondria there, right? Okay. We are zooming in right here. Okay. Now, embedded in the cristae, we're going to have a bunch of membrane proteins. Okay, so these membrane proteins, it forms our electron transport chain. Okay? What is the ETC anyways? What does it do, right? As the name implies, the electron transport chain transports electron. Okay, you put an electron on it, then it will pass on to the next one and then to the next one. Okay. Remember, in the matrix, that is where step number 2 and 3 took place, right? Number 2 is the pyruvate oxidation. Number 3 is the Krebs cycle. If you look at the chart that we filled in the study guide, you will notice in step 2 and 3, we created a lot of NADH and FADH, right? FADH2, right? So what's going to happen is the NADH will come to the first protein on the ETC. And let me be clear, right? Like on the cristae, there are like, thousands of these ETCs, right? For simplicity, I'm only showing you one of them. Okay, so the NADH will come to the ETC. It will release the electron onto the ETC, and afterwards it will become NAD plus again, right? Okay, it dropped off the passenger. Now it's ready to pick up another one. So this will go back to maybe pyruvate oxidation, it go back to Krebs cycle where it will pick up more electron and then just keeps on repeating this process. All right. So this electron, very high in energy, all right, it's going to get passed down the ETC like a hot potato. As the electron goes down the ETC, the energy from that electron will be harnessed, and we will use that energy to pump H plus from the matrix into the intermembrane space. Okay, so the energy from the high energy electron will be used to pump H plus into the intermembrane space. Over time, if this keeps on happening with more NADH coming in, you will create a high concentration of H plus in the intermembrane space, where you will have a low H plus concentration. Okay, as we learn from transport, we know things will go from high concentration to low concentration, right? But it's not going to be able to come back in through the membrane because the membrane does not allow charged particle to go through, right? Due to the hydrophobic nature of the phospholipid bilayer. So the only way for the H plus to come back in is through a special protein. Yeah, that looks like a little doorknob. It actually does. And then these H plus will go through this special doorknob protein. This is called the ATPase, okay, or ATP synthase, depending what you read, doesn't really matter. And what happens is, as the H plus comes back in through this special enzyme, this will spin, okay, like water through a turbine, and that will allow the ADP to pick up a phosphate, right? And then using this spinning mechanism, it will create ATP. Okay, so these ATP are created using these high energy electrons, right? Okay, that's why it's called oxidative phosphorylation. Questions? Any questions? No questions. Wonderful. Okay, so there is one last thing that we'll talk about. This electron, and this is very important, okay? There is going to be some application question related to this concept, so make sure you understand. So the electron will get passed down, okay? And eventually at the end here, something needs to take the electron away, okay? Otherwise, if the electron gets stuck here, the next one cannot slide, cannot come down, okay? I think of like my kids at the park, right? Okay, like take them to the park and then they go on the slide. There's always that kid at the bottom, okay? Sometimes they would just like linger there, like they just stay there and don't go away. But so, you know, the next kid can't go unless this one is like taken away, right? So the parent will come along, okay? And then it will take the kid away, and then the next one can go, right? So the same thing for the electron. The electron, if it's stuck at the bottom, the next electron can go down. And if the next electron cannot go down, we cannot pump H plus, right? And then we cannot make ATP. So something, the parent must come and take the electron away, okay? So what molecule is responsible for removing the electron from the ETC? Anybody know? Yes, Tyler, oxygen. Very good. So oxygen will come and it will take the electron away. And in the process, right, the oxygen gains the electron. It is going to be reduced when you're gaining electron, and that creates H2O. All right, so that's why we call oxygen O2 is the final electron acceptor. Okay, if we don't have the oxygen, you're not going to be able to remove the electron, and this whole mechanism won't work. We won't make ATP. That's why if we don't breathe in oxygen, we die. Okay, because we stop producing energy, and nothing works without ATP. Okay, so that's what the ETC does. All right. Okay, let's do another poll just to keep you guys engaged. Go ahead. Oh, sorry. I launched the wrong poll. I always do this. My bad. This is multiple answers. Okay. Let's try that again. Sorry. All right, that has been a minute. I'm gonna end the poll, and you know, you just have to look at the chart that I wrote you. Right? Look at the stars. Right? So there's a red star. There is a red star here. Those are the places where ATP are produced by substrate level phosphorylation. Right? Okay. No ATP is produced in the second step, and everything in the last step is produced through oxidative phosphorylation. So therefore, the answers are this one and this one. Sorry, this one and this one. First and the third. Okay. I actually got a lot of email from people saying how can I improve on my quiz? You know, how like a lot of questions. Like it seems like I don't know where to find the answer. A lot of the answers are actually in the study guide, right? Okay, so like after class, you should maybe print another fresh copy and do it yourself, right? Put it in your own words, right? And all these little subscript, all these extra notes are important, right? When it comes to answering the questions. Okay, what's the next one? Can we do the next one? Yeah, we can. Let's try that. Okay, so first one, NAD plus has more chemical energy than NADH. That's not true. Okay, after you pick up the electron, after NAD plus picks up the electron, it gets more energy. So this one has more energy. This should be less. NADH has more chemical energy than FADH2. Yes, we talk about this. NADH has the enough energy to make up to 3 ATP, whereas this one can make only up to 2, right? And NAD plus can be oxidized into NADH. At first glance, this seems logical, right? Like NAD plus becomes NADH, right? But you must think about how does NAD plus become NADH? Okay, in order for NAD plus to become NADH, it needs to pick up an electron, right? And if you are gaining electron, that is not oxidized. Okay, this is not oxidation; it is reduction, right? So NAD plus can be reduced into NADH. Okay, that's why this is not the correct answer. Okay, glycolysis can still take place without NAD plus. So that's not true because you need to have the NAD plus to take away the electron, right? If NAD plus is not available, then you'll be stuck after the energy investing phase, and it won't go on. Okay, so let's come back to this in a bit. We will skip the fermentation stuff for now. We will fill in this chart first. Okay? The reason I put this on here before this one when I type it up is because that's the only thing that would fit before the Word document starts a new page. So I just try to save space. In any case, let's try to do number 4 right here. Okay. So this will help us account for the 36 ATP that will be produced. Okay, and just to reiterate, 1 NADH equals to 3 ATP, right? Under ideal condition. Okay, does it always give you 3? Probably not. Okay, there's probably some efficiency thing going on that would reduce the maximum output, but under ideal condition, 3 ATP, whereas 1 FADH2, right, under ideal condition will be 2 ATP. Okay, and I just want to go back to here and add on a little bit here. The NADH, right, will deposit the electron at the first protein in the ETC. So in other words, what that means is by the time the electron slides through the ETC, it will have pumped enough H plus such that when they do come back down, you will produce 3 ATP, right? Okay. Whereas FADH2, because it has less chemical energy, it will be depositing the electron at a lower point. Okay. So when the electron is deposited at the second protein, it will only pump, enough H plus to make up to 2 ATP. So because the starting point is lower on the ETC, FADH2 yields less ATP. Okay. So here, let's keep track of our numbers. Number of ATP through substrate level phosphorylation. So in glycolysis, right? In glycolysis, there will be 2 net ATP. Remember, you invested 2, and then you produce 4 in the yielding phase, right? So 4 minus 2 gives you 2 net ATP. Okay. Energy carrier produced. Okay. If you flip back to the first page where we fill in the chart, you will notice we produce 2 NADH. Okay. Now there's a little bit of a caveat here. We get a little bit of a catch here. These NADH are produced in the cytoplasm. Okay. Remember, glycolysis happens in the cytoplasm. These happens in the matrix, right? Okay. So let me just redraw this for you. You don't have to redraw. You can just watch. Okay. So in the glycolysis, the NADH that you produce will be in the cytoplasm, and there are 2 NADH from the glucose. Now, in order to get to the ETC, it must go into the matrix, right? Okay, but this molecule is quite big, and it's not going to be able to, get past the double membrane. So what happens is it will actually just give off the electron. Okay, and then the electron will be passed on to an FAD. Okay, so the FAD will become FADH2. Okay, so the energy carrier does not go through, only the electron does. Okay, and every time you have like a transaction going on in the body, it causes energy. Okay, so that's why it's not going to be another NAD plus picking it up. Okay, you're going to have to spend a bit of energy just to do that. And that's why the energy becomes lower. Okay, so these 2 NADH that you produce in the glycolysis will actually be, the electrons anyways, will be transferred to the FADH2. Does that make any sense? Any follow-up question? Okay. Don't give up on me yet, okay? Like some of you, I feel like you might be thinking, oh, this is too complicated, just throwing in the towel. Okay, if you do need clarification, please let me know. Okay, so how many ATP are we going to be produced at the ETC? Remember, this is through oxidative phosphorylation. Okay, so because we have 2 FADH2, right? So 2 FADH2 multiplied by 2 ATP, that's going to give me 4 ATP, right? So total number of ATP from glycolysis alone, we have 2 net plus 4, so that's 6 ATP. Okay, I never know whether to put an S in the end or not. It doesn't really matter. 6 ATP. Okay, pyruvate oxidation. Any ATP from pyruvate oxidation? How many? Anybody? You flip through your notes, look at the chart. How many? That's right, none is correct. None. Right. Energy carrier per glucose will produce 2 NADH. Okay, now this time we don't have to transfer it to a FADH2 because we are already in the matrix, right? So 2 NADH multiplied by 3 ATP will give me 6 ATP. Okay, so from pyruvate oxidation, each glucose will get 6 ATP. Okay, Krebs cycle. Krebs cycle, how many ATPs from substrate level phosphorylation in the Krebs cycle? Look at the little circle thing I draw and a bunch of stuff coming out from it. Okay, in a way, you guys are a little bit fortunate, right? Like you can have a little, help sheet on the test. You don't really have to memorize all these things. If you're doing the test in class, you would have to. So how many ATP from Krebs cycle? Yeah, Aurora said 2, Tyler said 1. Yeah, 1 per spin, but 2 per glucose, right? Okay, and we're talking about per glucose here. So that's 2 ATP. And energy carrier, we have a bunch of energy carrier. 6 NADH and 2 FADH2. Okay, so we're gonna have 6 NADH DH times 3 ATP. That gives you 18. And we have 2 FADH2 times 2 ATP. So that's 18 plus 4, right? Okay, so 18 plus 4 is going to give me the 22 ATP. Okay, so 22 plus 2, 24. So we go 6 plus 6, which is 12. 12 plus 24, that's the 36 ATP. Okay, and you can see here, right? Like only 4 of them are produced through substrate level phosphorylation, right? So like if you take a calculator and you do like 4 divided by 36, 4 divided by 36, that's approximately 11.11%, right? That's what I said earlier. Only a small percentage of ATPs are produced through substrate level phosphorylation. Okay. I'm going to let you sit on that for a bit. Let's take a 5-minute break and we will resume when we come back. Okay, so 9:35-ish. Okay, I'll see you in a bit. Hello, it's me again. Let's keep going. Oh, there is hold on, there's a message. Who's this? Okay, let's see. There we have a poll question. Go ahead. Okay, is that all? Anyone else participating? I was gonna end the poll here. Okay. So this is quite easy question. Here we have 4 NADH. All we have to do is multiply by 3, right? Okay, so 4 times 3 is 12. And for FADH2, we multiply by 2. Because each of these can produce 2 ATPs. Each of these can produce 3 ATPs. So you add them together, 12 plus 6 is going to be 18. Okay, not sure like how some of you chose the other ones. Anybody like does it make sense now, or like do you want me to clarify more? like more than one of you chose 21 and 17. I want to know what the confusion is so that I can help you address it. Maybe send me a message or something. Okay, let's try another one. Okay, this is a little bit harder. It's not a multiple choice, but if you have the answer, please please type it out in the chat box. Okay. I will give you a couple minutes to do this. Okay, we have people say 120. Oh, Tyler, I mean like all the NADH and all the FAD, all of them considered together, not just the substrate level phosphorylation. Okay. A couple people got 120. Just gonna weigh out a little bit. Okay, more 120. Anyone not got 120? Okay. Let's okay. Will someone say 60? Yeah, let's see what the answer is, okay? Oh my goodness, it's not erasing. Give me a second. There we go. All right. So let's talk about one turn. Okay, what's going on with one turn? All right, you can look at that the thing we wrote, right? This chicken scratch right here. One turn, we will have two, forget the carbon dioxide is important. We'll have 3 NADH, 1 FADH, and then 1 ATP, right? Okay, so let's write that down. One turn we have 3 NADH, 1 FADH2, and 1 ATP. That's one term. Okay. So you can do this in one of two ways. We can calculate the number of ATPs produced per turn. So, you know, this can be times 3, times 2, and then this is just as is. So that will be 9, 2, and 1. Right? So 12 ATPs for one turn. Are we okay so far? Where these numbers came from? You know, how do I get the 12? Yeah. Okay. So can you explain once more time? Okay. Now these numbers, the 3 and the 1 and the 1, that's just given based on, you know, what I wrote here. Okay, there's no like explanation to it, if you would. Okay, for one turn it is just, you know, 3 and 1 and 1. You mean how do I get the 3 and the 2 here? Is that your question, Adriana? Okay, it's always 3 for NADH because, you know, we roll it down, right? In here, one NADH can produce up to 3 ATP, whereas FADH2 will produce up to 2 ATP, right? So NADH will always times 3, and then this is always times 2. Where are we? Right here. So we okay? We got the 12, yeah? Then we're gonna do it for 5 turns. 5 turns, so times 5. Times 5, right? So you can just, you know, times the ATP by 5. 5 times 12 is 60, I think. So that's 60 ATP. Okay. Now I can kind of guess why a lot of you said 120, because you're thinking, well, I thought you said the Krebs cycle spin twice per glucose, right? I'm not asking about per glucose, right? I'm just asking about 5 turns of Krebs cycle, right? Okay. When would the answer be 120? The answer will be 120 if I say there is 5 glucose. If you're going to metabolize 5 glucose, how many ATP will be produced from Krebs cycle alone? Then the answer will be 120. Do you understand the distinction? All right. Yeah, I mean, we can try this one as well. Go ahead. Okay, so the correct answer here is B. When something is more electronegative, it means it has a higher attraction to electron. And that's what you need, right? If you think about it, the ETC, each protein has to be more electronegative than the previous one. That's how it is possible to cause the electron to go forward, right? So, you know, if this concept is a little bit abstract, you can think about like a magnet, okay? So you know, you have a little metal ball here or something. In order for the ball to move on, the next magnet has to be a stronger magnet than the previous one, right? Okay. And when you get to the last one, oxygen would be like the strongest magnet to pull off that electron. Okay. This is the one with the highest electronegativity. Did you guys actually talk about electronegativity in chemistry class? Just wondering. Or is this like a new concept to you guys? No electronegativity. Okay, so I'll just write it down for you. Electronegativity means it's a measurement of attraction to electron. Okay, high electronegativity equals to high attraction. Okay, so if something is very electronegative, it would have a very strong pull on the electron. All right. Okay, so we have some other questions here. We will get to it in a bit. Let's finish filling in this thing. Okay, let's go back to the first page. We have lactic acid fermentation and alcoholic fermentation. Let's write this down again. We start with glucose, and then we have the 2 pyruvate. Okay, so where do the pyruvate go? Well, if you have enough, if you have I guess we'll just say enough. If you have enough O2, okay, if you have sufficient O2 supply, then you will do the stuff that we talk about, okay, like the pyruvate oxidation. We will do the Krebs cycle, okay, and then we'll do the ETC. Okay, so this branch, which is what we have been talking about for the past, whatever, hour and a half. Okay, this is aerobic respiration. Okay, it requires oxygen. If we don't have enough oxygen, okay, O2 is not enough, lack of oxygen, then we will do fermentation. Okay, so fermentation is an alternate pathway. It is called anaerobic respiration. Okay, so there are 2 types of fermentation that exist. Lactic acid fermentation, that's the one that we do. Alcoholic fermentation, that's the one that things like yeast and bacteria do. Okay, so in lactic acid fermentation, that would be like muscle cells. Okay, if you are running as fast as you can, after a minute or 2 minutes, then your thighs might start to burn a little bit, and then that burning sensation, it's a message that your muscle cell is telling you, hey, we are running on fermentation right now. Okay, you're not supplying enough oxygen, so we're going to switch to fermentation. Okay, so essentially, it happens when O2 demand exceeds O2 supply. Where does this happen in the cell? It happens in the cytoplasm. Okay, and we will generate lactic acid or lactate. And the most important thing is to regenerate NAD+. Okay, and I'll explain what that means in a bit. We'll just fill this in first. Alcoholic fermentation would be things like yeast, right? Some bacteria. When does it take place? Pretty much all the time. Okay, they lack the machinery to do all these like Krebs cycle, etc. They don't have that kind of stuff. Okay, so they rely on fermentation to get their ATP. Where in the cell, also cytoplasm. And the final product would be carbon dioxide. Right? So if you bake with yeast, the carbon dioxide helps your dough rise. It will create ethanol as well. So that's how we make drinking alcohol through fermentation. And it will also create NAD+. Okay. Moving on. We'll come back to this. Let's fill in the chart here, the diagram here. Okay, so on the left, we have the alcoholic fermentation, alcohol fermentation, or ethanol fermentation. And we'll go through the glycolysis process again. So we have the glucose here, and this is glycolysis. Okay. Now in glycolysis, in order for this to take place, we're going to need to have 2 NAD+, right? The NAD+ will pick up the electron. It will be reduced into NADH. Okay, never mind this 2, like 2H+. We didn't really talk about it. So, we're just going to cross that out. And of course, glycolysis will produce 2 ATP, right? Through substrate level phosphorylation, and you will get pyruvate. Okay, so under normal circumstances, the pyruvate should go on to do the step 2, step 3, and step 4. Okay, but we are talking about like a simple organism, like a yeast, like bacteria. They don't have those complicated proteins. They cannot do anything related to the Krebs cycle. So instead, what happens is the pyruvate will get converted into something called acetaldehyde. Okay, that's what this thing is called. You will learn about this in organic chemistry next semester. And the acetaldehyde is going to get converted into ethanol. Okay. Again, ethanol is the drinkable alcohol, right? Not all the alcohols can be consumed, right? Like rubbing alcohol is the one that we all buy during the pandemic. Those are propanol. You cannot drink propanol; it's poisonous. Okay, and then there is the methanol as well, right? Methanol also poisonous if you drink it. Okay, you go blind and then you will die. Very sad. So don't do it. Okay, so that is that. Okay, so the whole purpose of this fermentation is to give you a place to deposit those electrons and regenerate NAD+. As I said before, if you run out of NAD+, then this will not happen, right? And if this doesn't happen, you will not even produce the 2 ATPs. Okay. So for a simple organism, that's their sole source of ATP, right? That's why they need to regenerate all these things. Okay, so I'm gonna write this down here. The purpose of fermentation, doesn't matter which one, okay, both kind. The purpose of fermentation is to break down pyruvate and regenerate NAD+. Okay. Such that glycolysis can continue. Okay. All right, so what happens for us? Well, same thing. This is glycolysis again. And in order for that to happen, we're going to have the NAD+. It will pick up the electrons and it will become NADH. Never mind that. And you will get your 2 net ATP and you will get your pyruvate. Okay. 2 pyruvate, I should say. And in this case, we will pick up an electron from NAD+, regenerating NAD+ in the process, and the byproduct is lactate. Okay. For those of you who are interested, like lactic acid is when you have the extra H here, okay, and when you don't have the H, then it becomes lactate. Okay, that's all. But again, we're not going to make a distinction. Any question? Okay. For the interest of time, I'm just going to fill this in and you can check it yourself. later on in the when I post this online. Okay. Long answer questions. Any requests? Okay. If you thought of anything, we can take it up next class. I do want to talk about the application questions here, though. Okay, and let's start by doing a poll question. That's related to the application question. Go ahead. Only 3 choices. If you guess, you will have a 33% chance of guessing it right. Okay, that's been a minute and a half. Kind of half for A and B. So anybody want to explain their choice before I take it up? Nope. Okay. I will explain it then. So here is my ETC. Okay. So as you know, normally you will pump H plus into the intermembrane space, right? You'll pump H plus. Okay. Now, if we are poisoned by hydrogen cyanide, okay, so we have a CN here. Okay. It will bind to one of the membrane proteins and block the movement of electrons. Okay, so this is like the kid that won't go away, basically, right? So the electron will not be able to slide down the ETC because of the presence of cyanide. Okay, so if electrons are not being passed down, then the H plus will not be pumped into the intermembrane space, right? Okay, essentially having cyanide there will interrupt the transport of H plus from the matrix into the intermembrane space. Okay, so if this goes on, what happens to the pH here? Well, you will have a decrease in H plus because you're no longer pumping them in, and if you have less H plus, it is going to increase in pH. It is going to be more basic, okay, as opposed to more acidic. Okay, so the correct answer is going to be higher pH. Okay, does that make any sense? All right. Here is another one, but I don't think we have time for that. Let's just go back to this for a second. Here's another one. Speculate what would happen if a toxin disrupt the integrity of the inner mitochondrial membrane. Okay, so I'm just gonna go back to this diagram here. Okay, so forget about the CN for a second. Okay. If there is a bunch of holes in the inner membrane like this, a bunch of holes, then all the H plus that you pump over, right? Remember, you're gonna have high H plus over here, right? Okay, all that H plus, instead of going through the ATPase, they will just leak through the holes. Okay, they just leak through the holes. Okay, so if they're not going through the turbine, then you are not producing ATP. Okay, so in this case, right, you will have a reduction in ATP production. Okay, depending on how severe it is, it could be lethal. Okay, you could be like extremely tired by not having enough ATP. All right. Okay, I will post the answer for the fill in the blanks like I usually do, and then you can check it after you fill it in. Is there any question before I talk about the lab? Okay, great. Just to remind you one more time, a lot of you missed this at the beginning. Next week, there's no class, it's engagement week, okay? And then the week after, you're gonna have your test number 1. Test number 1 is lecture 1 to 5, okay? So everything we've done so far, including the lab, there will be questions about the lab as well, okay? And that's gonna be happening on the Wednesday, okay? Of course, it's gonna be available from 6 to midnight, essentially. I will post the information about the test later. And so to help you review for the test, our tutorial will be on the Monday on the 28th, okay, instead of the usual Wednesday, okay? Of course, I will send an email to remind you again, but please mark it down on your calendar. Okay, let's take a look at that lab of yours, because like I said, it will be there will be lab questions on the test as well. I mean, again, it was not done as well as I hope it would be. I have a feeling a lot of people did not watch the demo, and a lot of people just go on the internet and try to find answers, and it turned out not to be applicable to what we're doing. That's why it was not done correctly, and I feel like a lot of people maybe did it last minute, and so it was not the best quality. Anyways, I'm gonna go through some of these things. Make sure that you have the answer. I mean, you don't have to write it down. I'm going to be going through it quite quickly, but you can rewatch this segment later on in the recording if you need to. Okay, so the purpose of this experiment, right? I literally typed it out for you guys during the tutorial. It's to investigate the effects of temperature on membrane permeability, okay, on the beetroot membrane permeability. Hypothesis, you can say increasing temperature will increase the membrane permeability. You can say increasing temperature will decrease the permeability. You can say changing the temperature will have no effect on the permeability. Surprisingly, a lot of people say changing the temperature will not have an effect on the membrane permeability. It's surprising because I thought it would be common sense and based on what we learned in class, right? If we crank up the temperature, it's going to become more leaky, right? But in any case, if you got the wrong prediction, it's fine. Okay, as long as you roll the hypothesis, it's okay. Next one, dependent variable is the color intensity. Okay, you can say the absorbance, whereas the independent variable is going to be the temperature. This one was poorly done. A lot of people say because the beetroot has the pigment, which is true, but then like, why not these things, right? Like, make sure you address my question here, right? So number one, the pigment is very strong, okay, very vibrant. It's easy to detect visually, okay? Yes, carrot and tomatoes, they also have their own pigment, but it would take a lot of carrot to develop a very intense orange color, right? For example, so that's why carrot is not suitable, even though it has like beta carotene in it. Tomatoes, same problem with the pigment, but also it's very difficult to cut the tomatoes into uniform size, right? If you ever cut tomato, you would know, right? It's very seedy, right? It's very liquidy in some cases. It's very hard to cut. Certainly not as easy to cut as the beetroot. So for that reason, beetroot is the more appropriate choice. Okay. And some people ask, how are we supposed to know that? Well, not everything is going to be stated explicitly in the video, right? Like you're supposed to think a little bit about it yourself and, you know, do some research if you need to. Safety, for the most part, is fine. A lot of people lose mark because they just list a bunch of stuff like goggles, knives, gloves. What about knives? What about the goggles, right? Like you have to describe it a little bit. This one, the control is the 20 degree. A lot of people put 0 degree. Remember, the control is the one that is not being manipulated. Okay, so 20 degrees is the closest to the room temperature. And in this case, that would be used as a control. And the second part asks you, what's the significance of having a control, right? By having a control, it allows you to have a reference to compare your experimental group with, right? Okay, it gives you like a normal reference to base your observation on. Okay, some people also say there is no control in the experiment. So again, right, you need to have a control for the experiment, otherwise you have nothing to compare it to. Consideration, you have to make sure that they're the same size. You have to make sure that they have the same surface area, make sure they can fit in the test tube, for example. And that is important because that's part of your standardized variable, right? So that's the keyword I was looking for. Why is it necessary to rinse the cut beetroot? To rinse the beetroot, because when you're cutting it, the damaged cells will release the pigment. And if you just dip that into the test tube right away, then it will affect the color intensity, right? So by washing it off, again, it's part of the standardized variable. You're making sure that everything starts without any pigment on them, right? And those are the 2 marks that 2 points I was looking for the 2 marks. Here, in order to get 4 marks, you need to tell me, you know, if you leave it too short, then it might not be enough time to develop the color. And if it's too long, one of 2 things can happen. Number 1, the temperature will start to cool off. And number 2, if you leave them long enough, eventually the color might all be the same, right? Then the 40 degree is going to end up being just like the other ones, and you would observe no difference, okay? So that's that. Correlation, again, I'm just looking for very simple. As the temperature increases, the color intensity also increases. And then number 2, many people missed part B. What is qualitative analysis, okay? Qualitative just means you are making observation with your senses. So in this case, you're making the observation using your sight. You're trying to see, you know, the color intensity. This is darker than that, which is darker than that. Right? It's purely based on senses. There is no numeric measure. Okay. Some people say qualitative analysis means it cannot be measured by a number. That's not necessarily true, right? We can actually measure the color intensity using a number, and in fact, we did in the lab, right? But it's just that, you know, you're using your senses instead of using numbers. How were the results measured quantitatively? We use the colorimeter, right? Okay, many people put color emitter. That's not correct. It's colorimeter, but I did not take off the marks. Okay, it's probably, I don't know, maybe it popped up in the auto generated caption or something in the video. Like many people put color emitter. Okay, I'm not sure where that came from. But I mean, I know you guys have like a WhatsApp group or something. So maybe one person said color emitter, and then whoever's in that group decided to use that as well. So it's a calorimeter, okay? In the comments of your lab, you will see, I would have wrote that for you. The relationship measured by absorbance by the calorimeter and the color intensity. Okay, basically, the higher the color intensity, the higher the absorbance. That's all I'm looking for. The graph, most of it was well done. You know, if you have your two axes label, you make sure you put the absorbance on the Y and the temperature on the X, right? And then you get the line. You will get 4 marks. You will lose mark for not labeling the axes. You will lose mark for, you know, creating the wrong graph. Some people graph like 2 lines instead of absorbance versus temperature. You would have loose marks for that as well. At what temperature does the absorbance peak? I would accept answers anywhere between 60 to 80 degree. You will see the graph kind of go sideways, and then, you know, there is the sharp increase, and then it kind of plateaus a little bit, right? So peak absorbance occurs at about 60 to 80. I know 80 is more intense than 60, but then there is not like a sharp change, right, between 60 to 80. So, you know, any temperature between that would be okay. I would not expect the absorbance to peak at the same temperature for other plant cells. A lot of people say, well, the red cabbage also has the same pigment, so I would expect them to peak at the same time. The peak temperature has nothing to do with the pigments, okay? The peak temperature has to do with the composition of the membrane, right? Does it have the same amount of cholesterol? Does it have the same amount of, you know, saturated versus unsaturated fatty acids, right? All those factors are going to determine the fluidity of your membrane. And you know, let's say it has more saturated fatty acid, right? Then it would take a higher temperature in order for it to become more leaky, right? Remember the question we did in class during the tutorial, where I asked you about plants that are in colder environment and hotter environment, right? There are different cholesterol composition, for example, that would alter its permeability. Okay, so same thing here, right? Cabbage is likely to have a different composition than beetroot. Therefore, the peak temperature, the peak absorbance would be observed probably at a different temperature. 2 reasons to explain how temperature affects membrane. Number 1, at the initial rise, when you increase the temperature, the phospholipid starts to move a little bit faster. That creates a little bit of a gap between the phospholipids, and that allows some of the color to start coming out, okay? But for the most part, the membrane is still relatively intact at this point, okay? If you continue to heat it up, right, the second reason is at higher temperature, the protein starts to denature, okay? And then the entire membrane starts to fall apart, and that's when you start to see a lot of the colors coming out. Okay, so those are 2 reasons. A lot of people got the first one, but I forgot to mention about the proteins. What conclusion? What is the conclusion of the experiment? The conclusion is that temperature will increase the permeability of the membrane. You will lose mark if you say temperature will affect the permeability of the membrane. Yes, it will affect it, but you have to say how, right? Okay, and then do you accept or reject your hypothesis, right? So you either agree with it or disagree with it. I do want to point out one thing. A lot of people say the data prove the hypothesis. We never use that word in science, okay? It's not about proving hypothesis, right? You would say the data supports your hypothesis, okay? We can't really prove something. We can just, you know, find more data to support a hypothesis. And if it doesn't support it, then you will reject it. Finally, the last part here, it's what we talked about in class, right? When you freeze the beetroot, the ice crystal and the expansion of the water will rupture the membrane. Okay, so even when you bring it back to room temperature, the whole membrane is destroyed. So of course, all the color is gonna leak out, right? So that's why even if you leave it at the, you put it in water that is at room temperature, the absorbance is very high as what you would expect when you put it in, like, you know, 70 degrees water. Okay, that's the reason. So yeah, that's the lab. And, you know, we still have one more lab later on in the course. So if you didn't do so well this time, hopefully, you know, the comments and taking it up would help you do better next time. All right. So that's it for today. Remember, no class next week. But if you need help when you're studying, feel free to email me. I will also be posting a review sheet for you to do. And that's the review sheet that I'll take up during the test review on the 28th. All right. So yeah, thanks for coming and enjoy your week. Have a good one, guys. Bye for now.